Very few words are needed about Fred, just judging from the size of the crowd here. So I looked at Fred's bio and I thought about ourselves and I said, Fred's Executive Vice President and Deputy Director. We share that title. And then I looked at Fred and we're about the same height and we both have a really excellent hair. And then I got to the point where Fred had 900 publications and I realized that there was a significant difference between the center's two deputy directors. And so it's really a pleasure to introduce Fred. Fred is really just an incredibly important part of the Hutch since I've been here. I've known Fred for more than 25 years from when I came here initially as a fellow and then as a junior faculty member when Fred was division head. And Fred was my first attending and my first mentoring attending, and so it's had a very important part in my career development as a mentor here. And there's really nobody at the Hutch, in my opinion, or anybody that I know who has a broader sense of both the medicine side of cancer and the biology side of cancer. And Fred's scope of knowledge is just truly remarkable. He's had very important roles nationally. I can start to read through them, but it seems sort of silly, but they involve things like chairman of the Board of Scientific Advisors at the NCI, board member of virtually every oncology and hematology and transplant society that you can think of. He's a member of the National Academy of Medicine. He has, as I said, over 900 scientific articles. And as a result of all of this external activity, Fred is very much the external face of the Hutch in the cancer community. And I can tell you, whenever I go anywhere, 9 out of 10 times the first question I get is, what's Fred up to? And so, Fred has been such an important part of the Hutch's image across the world in cancer and beyond that we're all incredibly indebted to the role that he's played here. So I am really looking forward to learning about bone marrow transplant. And I think it's going to be a great talk for all of us. So welcome.
Sure. Great. Well, thank you very much, Bruce, and for that very generous and warm introduction, and thank you all for being here. I certainly hope that you enjoy the next hour or so as I tell you a story, and the story is the story of bone marrow transplantation and how it has grown from an idea to a life-saving therapy, to something that then gave birth to this new revolution in immunotherapy that is already curing patients and promises to cure many more in the future. So the story really does start with the atomic bomb blast in Hiroshima and Nagasaki, when thousands of individuals were killed by the percussion and heat of the blast. But then, over the next several weeks, other individuals started showing up at the makeshift first aid tents around the towns with fever, bleeding, pale, which we now understand to be radiation poisoning, but at the time the biology of that was really not well understood. And so after World War II, people became quite interested in trying to understand the genesis of this disorder. And so experiment was performed by Leon Jacobson at the Armed Forces Radiation Research Institute outside of Chicago. And what they did, what Dr. Jacobson did, was to take a mouse and to radiate it, and to simply, if you gave the right dose of radiation, you would get the same effect that happened to the victims of the atomic bomb blast. That is, they would develop what we now know to be bone marrow failure, with bleeding and infection and die. But if you took the spleen of the mouse, and see if we can get a pointer here, took the spleen of the mouse and put it in a lead box. So you exteriorize the spleen and you put it in this lead shielding, and then radiate the mouse would recover. And we now understand that in mice, the bone marrow and the spleen are both organs that contain what we term hematopoietic stem cells. So these are the cells that live in your bone marrow and have the responsibility throughout your life of reproducing and making all the red cells, the white cells, and the platelets that are in your peripheral blood. The red cells, of course, carry oxygen from your lungs to muscle so that you can oxidize sugars and give you energy. The white cells fight infection, and the platelets keep you from bleeding. And while you're sitting here, you're going to be producing billions of those cells to replace other cells that are dying, and these are all produced in the bone marrow, or in the case of the mouse, also the spleen. Now, this paper was published in the late 1940s, 1949 to be exact, and there was a guy by the name of Don Thomas, who was a chief resident at the time at Brigham in Boston, and happened to read the paper, and it struck Don that if you could take a mouse and radiate it and destroy its bone marrow and then give it a new bone marrow, what if the mouse had a disease in the bone marrow and you radiated it and got rid of the diseased bone marrow and gave it a new bone marrow? Could you cure the mouse's bone marrow disease? And could you, of course, apply this to humans? And could this be a technique that you could actually use to cure virtually every disease that starts in the bone marrow? So that would be all hematopoietic diseases, those diseases where the bone marrow simply fails, like aplastic anemia, or where you're born with an abnormal bone marrow genetically, like sickle cell anemia, or where the bone marrow becomes malignant, like in leukemia and lymphoma. Don thought big, and he wanted to see if this was possible. And so after completing his residency in Brigham, at the Brigham, and his fellowship, he then moved north moved slightly south to Cooperstown, New York, to the Imogene Bassett Hospital, where he joined two other investigators, the guy with the bow tie, Joe Farabee, and Hal Lockley there. And they started having this idea of trying to take this concept from a theory and use it to actually cure patients. And they started to work in animal models, and they had some success in animal models. And then in the 1950s, they happened upon a patient. And this was a young girl who was 4 years old, who had leukemia. And at that time, there was no therapy whatsoever that was effective for leukemia. And this individual also happened to have an identical twin. And so what the group did was they took the individual, they gave her total body radiation, and then took the bone marrow from the identical twin and infused it intravenously into this young girl. Now, it turns out that these bone marrow cells miraculously, when they're introduced intravenously, know where their home is, and they go through your arteries and veins, and when they get to the bone marrow space, they stop, and they set up shop there and do their job. And so this young girl was radiated, and if you see the bottom curve, where the white count is quite high, it goes down to almost nothing after the radiation. And then the new bone marrow starts to engraft, and the white count recovers. And the patient recovered in remission, which was quite dramatic. Unfortunately, by about 3 months later, the leukemia had come back. But it did prove the principle that perhaps this could actually work. Now, after trying it in the identical twin, they then tried it in siblings, brothers and sisters who were not identical twins. And in 6 straight cases, they failed. And so Don realized that there was something more to figure out. And so he stopped at that point trying to work it out in humans and went back into the laboratory. But other people didn't. And so after Don's first paper in 1958 showing the identical twins, other people around the world kept trying to do transplants, but they actually didn't understand all that was needed in selecting donors and how to do this. And so over the next several years, there were probably almost 200 cases of transplantation performed. And unfortunately, not one of these turned out to be a long-term survivor. And so by the mid-1960s, it was uncertain whether transplantation had any future whatsoever. And a lot of people left the field. Don, on the other hand, did not, and he came to Seattle. And then in the 1960s, he started working with a number of other individuals Shown here are Reiner Storb, a young Reiner Storb. This is in 1965, so you can do the math of how many years ago that was. So 53 years ago, there's Reiner. Bob Epstein, Dean Buckner. And they worked out the principles of doing transplants using a canine model. Now, the reason they used a canine model was because mice were inbred and the strains were not different enough to really recapitulate the kind of genetics that occurs in humans. Whereas dogs are an outbred species, they're large litter mates, and so you could get a litter of dogs and you would have essentially brothers and sisters. And they worked out that when they did transplants, sometimes in the litter it would work, and other times it wouldn't. And they hypothesized that there must be some kind of typing that could be done that could pick out the litter mate that actually was a match as opposed to the other ones that weren't. And so they worked that out and developed techniques to do what we now call HLA typing. In the dog, it was called DLA typing for the dog lymphocyte antigens. And they got to the point where they could repetitively pick out the litter mate that was a match. They could take an animal, radiate it, do the transplant from the right litter mate, and post-transplant give immune suppression to keep the new bone marrow from reacting in its new environment against the host, so-called graft versus host disease, and they could repetitively get successful long-term engraftment among matched litter mates. Whereas if they were mismatched and not DLA matched, the animals would all die. And so once Don had gotten to the point where they could get 100% success doing transplants between these matched litter mates, he felt it was time to go back into the human setting and began transplants for patients. And then, these are some of the dogs that were the long-term survivors. We used beagles at the time, mostly, for several reasons. Number one, they're very placid and very easy to deal with. Secondly, they're small enough that they don't require a lot of feed. Third, they have short hair, and so when you radiate them, they don't lose their hair. They just turn gray, as you can see. Many of these animals had turned gray. The long-haired animals, they would lose all their hair, and they would go from being a fairly large animal to a really small one, and it was tough. But you know, a lot of people have difficulty whether we should be doing experiments in dogs, and there was a lot of sensitivity to this, obviously, but it really was required to have a true outbred species, or this field would never have gotten started. And I will say that many of these animals Don actually brought home with him and kept them thereafter as pets on his house. So in 1970, it was ready to go back into the clinic, and they started doing transplants in patients who had end-stage leukemia. And this was a paper that I stumbled across that really set me on my career. By luck, I happened to read this paper. The idea that you could take a patient who had a bone marrow filled with leukemia, you could radiate that patient, you could take a normal bone marrow and replace it, and suddenly the patient would go back into remission, seemed to me like the most amazing thing that I had read in my life. And by the time I finished that article, I knew for myself what I wanted to do for my career. As you can see on your left, there's a bone marrow filled with leukemic cells. Every one of these cells looks alike. That's because they're clonal, all blast cells in the bone marrow. And then suddenly, a couple weeks later, after the transplant, you see a normal bone marrow with mature cells growing up. And you could actually occasionally cure patients who otherwise would die if you hadn't done this. Now, the first studies were tough. These were very, very sick patients. This is what's called a Kaplan-Meier curve. On the y-axis is the percent of patients surviving without leukemia. And on the x-axis here is the time. And what you can see is that when Don first started doing this work, within just a couple months of the transplant, over half the patients had died because they were so sick when they came in, had such end-stage leukemia. And to persist when that was happening really took a lot of determination, a lot of faith on the part of the staff, not just the physicians and the nurses that were facing such sick patients and having one after the other not succeed. But on the other hand, what you can see is with these end-stage leukemia patients, nonetheless, by the time you get out to 4 years, there still were 15% of them or so that were alive, free of disease, and that's 15 individuals who otherwise would have been dead. And it was seeing those long-term survivors, even though they were uncommon, that kept the team alive, kept the team together, kept the team working. And you know, this was one of the first patients who had ever been cured of end-stage leukemia. She went on and survived for 30 years after that initial transplant. That's what kept the group going. And with that insight that you could cure some of them, suddenly it became okay to start trying to do this a little earlier in patients' courses. And we took patients who had leukemia, who had not yet relapsed, just after they got their first chemotherapy. And this was the first group of patients that we transplanted while they're in first remission of their leukemia. And suddenly, instead of just curing a handful, suddenly the cure rates are getting up to 50 to 60%. In that, when we started seeing those kinds of cure rates, we realized that we really did have the potential to create a therapy that might have very important broad implications. So that's sort of the early history of transplantation. The people doing it went to incredible efforts to try and support those very sick patients early on. We all were platelet donors for the patients. Everyone knew their own HLA type, so we could be platelet donors for the patients that were there. To try and keep their white cell counts up, we would actually take granulocytes from patients in the community that had chronic granulomatous leukemia who came in and because they had too many white cells would donate their white cells. We put them in laminar airflow rooms so they'd be totally sterile, and we'd have to keep all their food sterile. We'd actually radiate the food because that was one way you could keep it sterile. And most foods are okay when you radiate them. They taste normally, but certain ones not so much. And so all the interns and residents had to taste the food before we give it to the patients. And I will tell you that radiated cheese is horrible. It breaks the double bonds and it gets rancid, so you don't want radiated cheese. We had to we tried to develop serum that could kill T cells, so-called ATG, and we actually inoculated our own horse with human T cells and had to take the serum out of that horse and then purify it and created our own ATG. So it was very primitive in many ways during those early days, but it did give rise to a therapy that now is incredibly important. So what do we do when we do a bone marrow transplant? Here you can see that there are within the bone marrow these so-called hematopoietic stem cells, pluripotent stem cells. So these are the cells that you have for your entire life that divide, reproduce themselves, but then also give rise to eventually the red cells, the platelets, the white cells, both granulocytes and lymphocytes that you will have for the rest of your life. And so any disease of the hematopoietic system can be theoretically cured with bone marrow transplantation. In addition, the bone marrow, because it is the site of making your lymphocytes and your granulocytes, is the home of your immune system. So presumably any immune disease could be cured with marrow transplantation. And so what I'm going to talk about next are the general indications for transplantation. And there are three big categories. One is actually fairly rare, but it is sometimes done, that there are certain diseases where you simply lack a gene product, like Gaucher's disease. And if you give enough, when you do a bone marrow transplant, you're transplanting about 10% of a person's cells, and so they can produce enough of the enzyme to cure Gaucher's disease. But those are uncommon indications for marrow transplantation. By far, the more common are to replace an abnormal lymphohematopoietic system. So again, if you are born with a deficiency or an abnormality in your hematopoietic system, like in sickle cell anemia or thalassemia, we can do a transplant and cure that. If you're born with a certain immune deficiency, that could be cured as well. And then we do transplants to treat malignancies, mostly hematopoietic malignancies. And this works in two ways. Number one, when we give chemotherapy, we generally kill the most rapidly dividing cells in the body. And the bone marrow are the most rapidly dividing cells in the body. And so the dose of any chemotherapeutic we can give is limited by bone marrow toxicity. And so if we can give a new bone marrow, we can give much, much higher doses of chemoradiotherapy than we would otherwise be able to give. And just by giving that dose escalation, by giving 4-fold more or 5-fold more chemotherapy or radiation than we'd otherwise be able to give, we can cure a number of cancers. In addition, as I'll explain shortly, there is a so-called adoptive immune effect. That is, when we give bone marrow from someone who isn't an identical twin to a patient, that new immune system can see the leukemia or lymphoma as being foreign and help reject it. And so we have both this dose response effect of transplantation, but we also have an immunotherapeutic effect of transplantation, and we'll talk a little bit more about that later in the talk. Well, why do we do transplants? We do it for a number of different diseases. These are some of the non-malignant diseases: aplastic anemia, hemoglobinopathies, congenital blood disorders, immunodeficiencies, autoimmune disorders. And to show you just how good we are getting with transplantation, these are the results that we have here. This comes from Reiner and our group. Our current patients, this is again the Kaplan-Meier curve. This is the percent surviving, and in the most recent cohort of patients, we're at 100%. 21 out of 21 patients cured of aplastic anemia with bone marrow transplantation. Now, before we had bone marrow transplantation, about 2/3 of these patients would be dead within the first 6 months after being diagnosed with aplastic anemia. So really quite an accomplishment. This is a little boy who was transplanted in 1980, and he is the first individual ever cured of thalassemia. Thalassemia is an inherited disease of hemoglobin, much like sickle cell anemia. There's about 250,000 patients born every year with thalassemia worldwide, most commonly in the Mediterranean areas. And by doing a transplant, we're able to cure thalassemia. And to show you the cure rate, this patient, by the way, was transplanted in Seattle in 1980. And these are the cure rates we now have for thalassemia. So we're just about at 88% survival, going out beyond 10 years for a disease that otherwise would be fatal in all these individuals. So quite an accomplishment. This is a paper that just was published. As I was stated, that if we can give a new immune system, we should be able to cure autoimmune diseases. Well, maybe we don't even have to give a new immune system. Maybe we just have to reset the patient's immune system. And so this is a very difficult disease called scleroderma. And what we show here is in a prospective randomized study that we helped lead. These are the overall survival in patients who had scleroderma, had their bone marrow stem cells taken out, and then given a high dose of systemic immunosuppression, Cytoxan, and ATG, and then given the stem cells back, resetting the immune system, and you can see this shows a significant survival advantage with the development of transplantation compared to what would happen if you didn't do that. And this is disease-free survival. And so, again, now this is the indicated therapy for certain autoimmune diseases, and a similar trial is ongoing right now for multiple sclerosis, which is another very devastating autoimmune disease. And so another indication for marrow transplantation. Turning now to malignant diseases, we have a series of malignant diseases which transplantation has been used for. And I'm just showing you representative studies. Here is a study where we published it quite a while ago, where we took patients who had acute myeloid leukemia, treated them to first remission, and if they happened to have a matched sibling, we did the transplant. And if they didn't, they continued to get standard chemotherapy, and you can show the dramatic differences in survival. And so ever since this was published, and now this is published over 30 years ago, transplantation for AML is indicated for patients who are in first remission with that disease. This shows you the transplantation for non-Hodgkin's lymphoma, patients who had been treated with conventional therapy, then they failed conventional therapy, and then they needed to be retreated, and they were either retreated with transplantation or with conventional salvage chemotherapy. And here you see this dramatic survival advantage with transplantation. And just turning to one more disease, this is multiple myeloma. This is another disease of the hematopoietic system where patients were treated either in a prospective, this is a prospective randomized way. They either got high-dose chemotherapy with transplantation as initial therapy, or they got conventional dose therapy. And you can see quite a bit of difference in their overall survival as you go out to 5 years with transplantation. And so transplantation is indicated for a large number of diseases, both of the non-malignant and malignant diseases as well. And because of that, the number of patients who are transplanted every year continues to grow. It now, particularly with the change in the socioeconomic status in China, has increased dramatically. And it's assumed, I shouldn't say assumed, it's estimated with fairly accurate data that there are probably around 100,000 transplants performed worldwide every year. 100,000. And that all began right here in Seattle with the pioneering work of Don Thomas. So that's quite an impact when you think about what our group has done. And that's, as I'll say again later, that's a contribution that is, certainly Don was the leader. There was a lot of scientists that worked with Don, but it was also the work of all the nurses, the technicians, the staff, the administrators that made that happen. And in 1990, early in the morning, Don got the call from Stockholm saying that he had won the Nobel Prize. And the first place that Don went that morning when he came into work was to the nurses' station to thank the nurses because for all the work that they had done to make it possible for what had been achieved. So I'm going to talk now for the next 25 minutes or so about what some of the major goals of our current research are. And I hope I'll be able to leave a couple of minutes at the end for any questions that you might have. But one of the things we'd really love to do is to be able to provide a safe allogeneic donor for everyone in need. A second is we'd like to eliminate transplant-related toxicities, make it a safer procedure. And finally, we would love to be able to harness this graft versus tumor effect that I'll talk about. Well, very briefly, HLA, human leukocyte antigen, is what determines whether you can accept a bone marrow transplant or not. It's on chromosome 6. It's codominantly expressed, and since you get one chromosome from dad and one chromosome from mom, that means that there are 4 possibilities, and so you can get this chromosome from dad, this one from mom, or this one from dad and that one from mom, or this one from dad, that one from mom, etc. So there's four possibilities of taking it that way, and so the chance that any 2 siblings will match is 25%. The average size of a family in the United States is now about, with the same parents, is about 2.2. And so overall, that means that any one patient has about a 30% chance of having a matched sibling. And that's what we found. So when I first came here, we would only do transplants on people who had identical twins or matched siblings. We would also only do them on anyone that was under age 40 because the procedure was so vigorous. But that left about 2/3 of people that did not have a donor. In the late 1970s, a patient came with, actually, a guy by the name of Bob Graves came with his daughter, who was the patient, and said, you know, my daughter has leukemia. I know nobody matches in the family, but I've actually been reading a little bit about HLA, and I know that there are different haplotypes that may be much more common than others. And I just happen to know my daughter has two of the most common haplotypes there are. Now, when I say haplotypes, what I'm talking about are these genes. There are many, many, many type A's. There are many, many B's. There are many, many DR3s. I'm talking about hundreds of these. And so there's thousands and thousands of possible combinations of HLA. But some are fairly common and some are quite rare. Bob Graves knew that his daughters were very common. And so he said, she needs a transplant. She doesn't have a match. And what I'm asking is, don't you think you could find an unrelated donor that happened to match her? Now, this had never been done. And so he talked to Don and talked to John Hansen, talked to a number of us, and we said, well, we'd see if we could find such a donor. We all knew our own HLA types, as I mentioned earlier, and so we went into our little files, and when I say files, I mean a 3x5 file card, which is how we kept the HLA types, and we found that there was a technician in the Hematopoietic lab on the 3rd floor that actually matched Laura. And so we talked about, could we do this? And we went to the IRB, and they said, sure, it seemed reasonable. And we performed that transplant in 1980, which was the first time transplantation from a matched unrelated donor to a patient with leukemia was performed, and John was the leader on that study. Now, the good news is that the graft worked well. There was no overt graft versus host disease. Unfortunately, Laura, while she went into remission, her leukemia came back within about 2 years, and eventually she died. But the family was so grateful for the attempt that they formed the Laura Graves Foundation, and eventually that morphed into what is now the National Marrow Donor Program, and this is now something that is worldwide. And worldwide, there are 27 million people that have agreed to be HLA typed and to serve as a donor for someone that they don't know. They're not doing it for any financial remuneration. I mean, you know, there's lots of good people in the world, and it's always wonderful to know that. Now, it turns out that with that kind of matching, we can find an 8 of 8 matched unrelated donor for about up to 70% of Caucasians. It's down to about 40%, 30 to 40% for Hispanics, only about 20% of blacks. Now, this is not because there aren't enough Hispanic or blacks in the registry. If there were more, it might help a little bit. But it's actually more what we understand is that human civilization began on the African continent. And so every possible HLA type is there. As individuals in, we don't know, 70, 100,000 years ago left Africa, they left in extended families. And of course, if they're an extended family, they only take a subtype of those HLA types with them. And so if you look, for example, in the Japanese population, there's a limited number of HLA types that are there that aren't represented in other ethnic groups. If you look at Ashkenazi Jews, the same thing. And so if you look at blacks, every HLA type is represented. So there's just more heterogeneity in HLA types. And so this would make it very difficult for us to find donors for blacks, Hispanics, and Caucasian. And this is one quick fact which I find amazing. If you look in the United States for all the searches that we start, and the patient is over age 50, we can find an 8 of 8 match for about 54%. If the patient is under age 20, we can find an 8 of 8 match for only 34%. So that tells you how dramatically the population of the United States is changing, how we're becoming a much more ethnically mixed group of individuals, and how there's more inter-ethnic marriages and kids, and you know, it's a tremendous thing, and there's a lot to celebrate about that, about how the country is changing. But for those of us who are looking for donors, it represents a little bit more of a challenge. So, what are we going to do about that? Well, the first thing is I'd like to tell the work about Effie Petersdorf, and what Effie has found is that you can actually find mismatches that are so-called permissive. That is, they allow one to get you can be mismatched at HLA-C or mismatched at HLA-DPB1 and still get a transplant without any increased risk of the graft being rejected or graft versus host disease. And not only she found that these permissive mismatches exist, but she's figured out why. It is because the genes that control HLA-C in these permissive mismatches don't express their protein nearly as much. And so they have low expression of HLA-C, and so you just don't see the mismatch. It's invisible to the incoming cells. Same thing with the HLA-DP. And so this now allows us to do transplants that can be single antigen mismatches. The second work I'd like to talk to you about comes from Colleen Delaney and Filippo Milano, who have led our cord blood transplant program. Well, it turns out that a cord blood, thing that attaches the infant to the mom, has about 100 cc's of blood in it. That's about a third of a can of Coke. But within that blood, it's very, very rich in hematopoietic stem cells. And we don't know the biologic reason why that would be so, but it just happens to be that way. And that's the infant's blood, not the mom's blood. And not only is it very rich in hematopoietic stem cells, but the T cells, the immune cells that are in there are very, very immature, and there's fewer of them. And so we're able to do transplants from cord blood that are mismatched up to 2 or 3 antigens with the donor and still allow the cords to take and not cause untoward graft versus host disease. And so with that, we just published this paper in the New England Journal that shows that our outcomes with cord blood transplants are every bit as good as they are with matched unrelated donors. And so now, with the capability of having 7 of 8 unrelated donors or cord blood, we can find donors for almost everyone in need. 95% of individuals, we can find either a cord or a single antigen mismatched or a matched unrelated for those individuals. And so we are getting pretty close to the point of having an allogeneic donor for everyone in need. Now, the next thing we wanted to do was make the transplant safer. This was a study that Ted Gooley in our group did, where we looked at the outcome of transplantation from 1993 to 1997. And this represents about 1,500 transplants done here in Seattle. And then we compared it to a group of patients transplanted a decade later, from 2003 to 2007. And this curve on your left shows the incidence of dying of a complication of the transplant. And as you can see, it has gone down from about 30% to 15%. So in that decade, we halved the risk of transplantation, and that translates directly into improved overall survival. Now, how did we do it? And this next little vignette just is going to tell you that so many different people are doing work here to try and make transplantation better. And oftentimes it's just a small, comparatively small step, but when you add them all together, it ends up being incredibly important for overall survival. And so previously, cytomegalovirus was a viral infection that caused a lot of deaths after transplantation. Between 1993 and 2003, we halved the risk of cytomegalovirus. How did we do that? It was the work of Joel Myers showing that there was an antiviral called ganciclovir that you could give prophylactically, and it would prevent most CMV deaths. Likewise, we decreased the risk of dying of a fungal infection. The HR means hazard ratio, and so after you do all the risk assessment, this says that the risk between 2003 and 2007 is 55% of what it was previously, so almost half. And again, how did we do that? This was the work of Raleigh Bowden and others, where we showed that if you give prophylactic fluconazole, you can decrease the risk of fungal infection with a transplant. Hyperbilirubinemia, that is liver disease, it went down by 75%, so we only had a 24% hazard ratio of having liver failure in the most recent decade. And how did we do that? Well, one of our fellows here in Seattle discovered that using ursodiol was effective in doing that, and he then did a prospective randomized study showing improved overall survival with this drug that protects the liver from damage during the transplant procedure. Acute graft versus host disease. So when we do the transplant, we're putting in a new immune system. That new immune system, if the patient is not getting a transplant from an identical twin, can see the patient as being foreign and react against the patient, and that can cause a skin rash, it can cause liver dysfunction, it can cause nausea and vomiting, and that can be mild, it can be severe, it can be fatal. And we have figured out ways that we can reduce the incidence of graft versus host disease by doing better HLA typing. And this is work that, again, came from F.E. Petersdorf and Claudio Anicetti and others, showing that we can reduce the risk of graft versus host disease by better donor selection in the most recent era. And finally, I just used respiratory failure here for talking about the overall damage to the patient that comes from the very high-dose preparative regimens, and we have shown that there are conditioning regimens that are not as intensive as what we initially used that can still get the job done, make sure that you get a graft, and make sure that that relapse does not occur. And so we've substituted certain drugs to make the conditioning regimens more tolerable. And so with all this, we have decreased the mortality of transplantation almost in half from 1993 to 2003. Well, with that history, then we said, well, what's happened most recently? And so I talked to Ted Gooley and George, and I said, well, we ought to look again. And so what we have found is this is the survival following transplantation from 1993 to 1997. And this red curve is 2003 to 2007. All those things that I talked about had been in place. And so now, when we look at the most recent data from 2013 to 2017, we see, again, continued improvement. Well, why is that? We've already taken care of CMV, we've already taken care of fungus, we've already taken care of having a better conditioning regimen, we've already made the better typing. And the truth is, we don't know. We're right in the midst of doing the analysis. I was here with George last night as we're slogging through these 1,000 patients so that we can do the right statistically sophisticated analysis to figure out why are we still improving, but we are. And that is clearly a very, very gratifying. So in the final couple minutes, what I want to talk about is transferring all of this excitement in bone marrow transplantation to something that may be even more important, that is harnessing the power of the immune system. This, again, is one of these Kaplan-Meier curves, which risk here, years here, and this shows the chance of a patient recurring with their malignancy after a transplant. And it's broken down to whether the patient got a twin donor or got a sibling donor who's not an identical twin, but did not develop any graft versus host disease, or a patient who gets graft versus host disease, acute or chronic, or both acute and chronic. And what you can see is that the relapse rate, the chance that the disease comes back after the transplant, is way, way higher if your donor is an identical twin than it is if your donor is a brother or sister. And not only that, but if your donor is a brother and sister, and we take the T cells out of the bone marrow, the risk of relapse goes right back up to what it would be if you had an identical twin. So this provides incredibly potent argument that when we do a transplant, it is the immune system that's coming from the donor that is seeing the leukemia as being foreign and helping to reject it. So that's a big part of why transplantation works. Now, once we realized that, we said, whoa, can we harness that? Can we find the cells that are reacting against the leukemia? Can we find the targets on the leukemia that the cells are reacting against? And can we segregate this graft versus leukemia effect from the graft versus host reaction? Because the graft versus host reaction causes a great deal of morbidity and potential mortality. And so just causing more graft versus host disease non-specifically is not a solution. And so this is the work of many people. I'm just highlighting here Hootie, Warren, Phil Greenberg, Stan Riddell, who have been part of a grant, the ALC grant that I've had for many years, where that has been one of the major goals, has been to do that. And what we have found over the years is that if you have a patient who's had a transplant and they recur and didn't have graft versus host disease, we can simply take lymphocytes from the donor, infuse them, and sometimes get that patient back into complete remission, but at the risk of graft versus host disease. Again, reemphasizing the fact that there is this GVL effect. So one of the goals then was, well, can we actually take cells from a donor and take cells that are individual against one particular target? And can we get those? Can we expand them? Can we give them? And can they function? And so Stan and Phil developed methods that early on in the early 1990s to take out T cells and to be able to clone them and expand them into large numbers. And then in an incredibly important paper in 1992, Stan, Phil, the group here, for the first time showed that you could take T cells from a donor, transfer them to antigen-specific T cells, so against one particular target, transfer them to a patient and have them work. And this was in the setting of CMV. Now, CMV is a virus that many of us have, probably 60% of us in this room had this when we were children. And it doesn't bother you unless you become very immune suppressed. If that happens, then it can blossom again and cause disease. In fact, it can cause fatalities after transplantation. And what Stan and Phil and others did was to take donors, take out their very T cells, isolate the ones that were against CMV, clone them into large numbers, and then post-transplant, infuse them to patients and show that you could reintroduce immunity to CMV, and the level of immunity was just the same as in the donor. So you could actually transfer these cells, they would persist and they would function, and you could protect patients from developing CMV. Well, once we did that against CMV, then the question was, can you do this for cancer? You can get the cells out, but can you find the target on a cancer cell? Can you expand the cells? Can you infuse them? And that has been, of course, the challenge that we've been working on for decades, to identify appropriate target antigens on leukemic cells, to engineer T cells to react specifically with those tumor targets, and then to assure once these cells are transferred that they persist, they expand, and they function. And that, of course, has been the work that you've been reading about so recently, about the so-called TCR T cells and the CAR T cells. And basically what we're doing is we're taking out T cells. They have their own receptor, but then we engineer them to have either a TCR, a T cell receptor in them, or to have an antigen, a chimeric antigen receptor in them that react against particular targets on the tumor cells and then infuse them into patients. And the ones that you've read the most about are the so-called CAR T cells against CD19, and the results there have been absolutely spectacular. I'm sure this will be the subject for another talk that you will hear. This is not my work. It is the work of many of my colleagues, and there's a very large story to be told here. I don't want to bore you, or I don't want to steal that thunder, but basically in relapsed refractory ALL, we are seeing a 93% complete response rate by just infusing one infusion of these CAR T cells to these patients who have refractory disease. 82% response rates in patients who have non-Hodgkin's lymphoma, and in CLL, we're seeing 85% complete response rates. And so this has been just an enormous success. The two products have already been approved by the FDA and are already out there saving lives. And there are many more to come. There are over 250 trials registered at clinicaltrial.gov about other CAR T cells against other targets that are on leukemia, lymphoma, myeloma, breast cancer, many, many different tumors. And this all arose from marrow transplantation, from Don's initial idea that we would be able to take a new hematopoietic system, put it into a patient, and save them from otherwise fatal diseases. This is, of course, Don had many, many accolades of being the father of marrow transplantation, but frankly, I think his greatest contribution is going to be looking forward to giving rise not just to transplantation, but to this entire field of adoptive immunotherapy. So the work I've talked about is that of many, many individuals, and I really can't tell you all the group that's contributed over the years. But again, I just want to emphasize that certainly the scientists have done an enormous amount here, but it is also the mid-level providers, the nurses, the pharmacists, the dietitians, the schedulers, the social workers, and other members of the SCCA UW Hutch family that have made all this possible. It really does take an enormous team effort, and I, for one, have been so blessed to be able to be a small part of it. So I'd like to thank you, and I think we have a few minutes to answer any questions that you might have.
We have this box to pass around. If anyone has any questions, you can toss it like a football or a soccer ball.
With the cord blood, how do you guys obtain the cord blood from the umbilical cord?
How do you get the cord blood?
Yeah.
So the mother gives consent beforehand. They're approached beforehand, and the whole idea is explained to them. And then one of the technicians actually has to go into the delivery room after the baby is safely delivered and the placenta has been delivered. Then the blood is simply drained from the cord and collected right there in the operating room. From there, it goes into the laboratory for further processing. It's not of any, if you did not collect the cord blood, that material would be basically placed in the garbage. I mean, it's not of any risk to the mother or the baby. It's after the whole procedure's been done. And it's about 100 cc's. Yeah.
Don't throw it at me on the I told you. I'd like to ask about GVHD. You've got, I'm assuming, you can anticipate some of the problems just during or right after transplant. But now with immunotherapy, they're starting to see, I believe, kind of unconventional, unanticipated GVHD like 2 or 3 years in the future. At least that's what I've read. So
No.
I don't know if that's true.
No, so GVHD, it's very interesting that GVHD, again, is this reaction of the donor cells against the host. And it comes in two forms: acute when it comes within the first 3 months, or chronic if it comes afterwards. It generally, though, it just takes time for the patient and their new stem cells to get to know each other and to develop tolerance. Now, I know that sounds sort of mystical, but the reality is, so this is just a little bit of Immunology 101. One of the big mysteries forever was how can you create T cell receptors or antibodies that can see everything? I mean, every bacteria that has ever existed, and even things that have never existed in evolution. If they then, in the future, your immune system could still see it. There isn't enough room in your DNA to code for all that. And so what your DNA does is there's a small segment where you make your T cell receptors and your B cell receptors, and it just rearranges in utero. and keeps reforming itself to form every possible shape. And so it's like taking the letters of the alphabet, and you can make a lot of words with only a limited number of letters, an infinite number, literally. And so that's what your body does, and that gives it all these different T cell and B cell receptors. So this incredible heterogeneity with just a little bit of DNA. Now that's a very clever way of making heterogeneity. There's only one problem. What's to say that those T cells won't react against yourself if it's a random process? And in fact, they do. And when you're an infant, about 70% of the T cells that you make are autoreactive. But you have a thymus. And so those T cells come around, and when they bind to the thymus, your thymus binds to the thymus, your thymus says, oh, you're not a good guy. And so it extinguishes those cells. Either they make them allergic or it gets rid of them. Now, when we do transplants, we're no longer infants in most of our patients, and our thymi aren't that effective, and so it takes time for the new body to extinguish all of these new reactive T cells that are against yourself. That just sometimes takes months, sometimes takes several years for that to happen. But eventually it does, and so you do get this tolerance that eventually develops. With some of the newer immune therapies that we're talking about, so there's two kinds. With some of the immune therapies, like the checkpoint inhibitors, who are just taking the brakes off your own immune system, we're allowing some of those cells to re-express themselves, those that were allergic, and they can start reacting against yourself. And we're finding some of the toxicities with the CAR T cells. We're just learning about some of those, but we've done a lot of work. Cam Turtle has been leading the march there for ways that we can decrease the toxicity that we see with some of those cells. Just talk loud.
So you were talking earlier
Okay.
you were talking earlier about how when the stem cell transplant, oh, it's a microphone. Oh.
That's what's there. It was.
Okay, great. Is this better? Great. Okay. So when the method of transplantation was discovered, you were talking about how when they inject the stem cells, they just kind of know where to go. Since the discovery and implementation of that process, what have we learned about how the stem cells are able to navigate their way just to make it there?
How they know where to go?
Yeah.
So basically, it's a system of integrins and selectins. So these are proteins that are specific for the cell and for the particular, it's like a zip code for the vessels. So that's how lymphocytes generally know where to go. They know how to go into a lymph node, or they know how to go to the bone marrow. And so it's like it's like a barcode when the cell. It's amazing to watch movies of this. You can actually see these. People have done these movies where you can see the white cells, the bone marrow cells after injection. They don't go down the middle of these. They're not in the middle of the lane. They're like going along the curb, and they just kind of roll down, and they keep traveling and rolling and rolling and rolling. And when they get to the site where they're supposed to be, it's like they go, whoa, I'm here, and they get activated, and they flatten out. Like a fried egg, and then they just migrate through the endothelium into their little niche. And to actually watch the movies of it is pretty fascinating. Early on, when they first did transplants, they did comparisons of trying to give the cells intravenously or arterially, or you could even give them intraperitoneally. So you can just inject them into the peritoneum. That actually works. The cells migrate and know how to get there. It just isn't quite as efficient if you give them intraperitoneally. There's not much difference between giving them arterially and through the vein. So we, since it's a lot easier to get into a vein than an artery, we give them intravenously.
So is that zip code sort of idea specific to the bone, or like?
What's that?
Is that zip code sort of idea specific to the bone region, or like how?
Bone marrow. That's another question. So bone marrow is actually can go everywhere. Can grow in virtually all your bones, but it likes the so-called cancellous bones. That is, the ones that are more central. And so bone marrow generally is in your vertebrae, in your hips. in your skull. It tends not to be in the long bones, unless, and you can actually do this, people have done these experiments, if you radiate all of the areas where you normally have cancellous bones, you can turn the other areas into bone marrow supporting areas. And so when you look at the bone marrow, like in a long bone, normally it just looks like fat. There's no cells there. But if you radiate the other areas, that yellow marrow will turn into red marrow with cells into it. So it has that capacity to do it. It's pretty fascinating. But this has been known for a long time. But yeah.
Thank you.
Yes, sure.
Good toss.
Guys.
I was wondering if we could apply what we've learned from marrow transplantation and the patient's immune response to organ transplantations.
So yeah, that's a very interesting question and something that's being done. One of the problems with kidney transplants, for example, is that if you get a kidney transplant, you generally have to be on immune suppression for the rest of your life. And even on immune suppression, there is a chance that you will eventually reject the kidney after a number of years. Now, if we could do a bone marrow transplant and give a new immune system to the person, and then you gave a kidney from the person who gave the bone marrow, the immune system of the kidney and the bone marrow would be the same. And you'd never reject the kidney. And so now there are ways of doing what's called micro transplants, where we give just a bit of the bone marrow from the kidney donor and some immunosuppression, and we're not looking to get full engraftment. We just want to get what's so-called mixed chimerism. We want to have some of those cells persist and stay, and then do a kidney transplant in that setting. And those experiments actually are ongoing in humans. There have now been at least, in the one experiment that I recently reviewed just last month, they had done about 25 combined bone marrow kidney transplants, and of the kidney transplant recipients, I think something like 21 of the 25 were already off all immune suppression with tolerance with a new kidney. It is really exciting. So again, you're exactly right that what we've learned about immunology and tolerance can have effects that we're not even thinking about, solid organ transplants. And if it works for a kidney, there's no reason it won't work for a liver. Now, for a kidney and liver, it's more, you can understand how that can be done because there's a lot of living kidney and living liver donors, particularly when a mom's, for example, donating liver to the kid, you just take a partial part of the liver and you can do that. So there's a readily available source of bone marrow. How to do that for the other organs is a little bit harder to imagine unless you have ways of being able to get some, for example, with a heart transplant. Of course, the victim, the donor, is someone that's died in usually an accident or something like that. So can you get marrow in time to be functional? But it's certainly possible to be done. It's being done now, to answer your question. Yeah.
No?
On that same note, if you had someone who had, let's say, 15 years ago gotten a kidney transplant.
I'm sorry?
If it's someone who has previously gotten an organ transplant, could you possibly now go back in and do a bone marrow transplant and see if you could help them?
So we actually published a paper. Chris Doney was the first author on it, where we looked at patients who had had solid organ transplants and developed leukemia and needed a transplant, and we were able to carry out bone marrow transplants without any difficulty in that setting. Now, could you, if someone had a kidney transplant from a donor and then had the same donor give bone marrow afterwards, would the patient then become tolerant? Yes, they would, and they would be able to get off all immune suppression. We've had other interesting experiments where you didn't plan it, but because of the coincidence of two diseases. The very first patient with sickle cell anemia that was ever cured of sickle cell anemia was here in Seattle, and it was a young boy who happened to have acute myeloid leukemia, who also had sickle cell disease. Well, we did the transplant for the AML, but in doing it for the AML, we cured the sickle cell disease. We had a group of patients that we recently reported that had Crohn's disease. Can be very difficult autoimmune disease of the GI system. And we had a group of 5 patients who had Crohn's disease who also had acute myeloid leukemia. We transplanted them for their leukemia. Their Crohn's disease was cured as well. So there are these accidents of nature that teach you a great deal about what can be done. And so, if we can make transplantation safe enough, we can certainly use it as a treatment for some of these other diseases as well. Okay.